This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM. This Week in Microbiology, episode 125, recorded on March 31st, 2016. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How are you?
I'm doing well, thank you.
Is winter gone from Ann Arbor?
It is. It's a lovely, rainy spring day here. 60s and kind of a gentle rain falling. Gives us hope for spring and green.
Do you have plants coming out of the ground there?
Oh, yeah. Yeah, little blue flowers, and the lawn is starting to get green.
Yeah, today we have 20 Celsius, although it is cloudy, but we have crocuses and gladiolas and all kinds of plants.
Nice.
Bursting forth. Yes, although, does it officially begin in April or is it March? I don't remember. When does spring begin?
Spring?
Yeah.
March 20th.
So we're in spring.
We are in spring.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Yeah, you have 80-degree weather there, right?
75, but it's going to rain. The April showers will begin tomorrow, and hopefully they will bring lots of May flowers.
Wonderful. All right, we are sponsored in this episode by the American Society for Microbiology, and they would like you to know about their Agar Art Contest. It's the second one, and here you take an agar plate and you make art using bacteria, fungi, whatever you like to grow on. Obviously, you need to have a quasi-lab at least to do this, and you take a picture of your piece and send it in, the picture that is, to ASM. The deadline is Friday, May 6th, and the winners and some runners-up will be showcased in a gallery at ASMMicrobe 2016 in Boston this June. You will also get a free ASM book of your choosing if you should win this contest. For full contest guidelines and terms and conditions, go to bit.ly/agarart2, the number 2 in all one word with agarart. It's a cool contest.
I think you said they're supposed to submit the photo, not the plate itself, right?
Yeah, I did.
Oh, yes.
We don't want the plates. That would be bad. People would freak out.
It would actually terrorize poor John Meyer.
Yes, John Meyer. We had great submissions last year as photos. Well, very creative stuff that people were able to do. And so we're looking forward to more. I mean, I am on the judging panel, so I benefit to see all these cool things. People really get into it. It's really neat. It's amazing what you can grow on a plate. As we'll see later, you can even make plates that are not even live. Okay, we have for you today a snippet and a paper. Unfortunately, Elio can't join us, but we will hold down the fort as best we can. And Michele will start off with a snippet from MSphere.
Yes, MSphere, and this article will be published April 5th, so it's in press. It's titled Culture-Independent Metagenomic Surveillance of Commercially Available Probiotics with High-Throughput Next-Generation Sequencing. And the authors are Jennifer Patro, Padmina Ramachandran, Tammy Barnaba, Mark Mammal, Jada Lewis, and Christopher Elkins, and they all work at the Division of Molecular Biology at the Center for Food Safety and Applied Nutrition at the U.S. Food and Drug Administration in Maryland. So as the title implies, they were interested in bringing state-of-the-art sequencing technology to study the accuracy of labeling in the probiotic industry. So as many of our listeners will know, probiotics are, according to the definition by the World Health Organization, live organisms which, when administered in adequate amounts, confer a health benefit on the host. So they were able to apply state-of-the-art next-generation sequencing to address the who is in the probiotic preparation, and then they combined it with a very traditional technology, plating for viable counts, to verify that, in fact, the microbes were alive. So their design was to select from the market 10 different commercial probiotic products. They isolated or analyzed 3 separate lots of each of these 10 products by, first of all, just extracting the total DNA, which they then used for whole genome sequencing. And then they took another sample from each of these 10 commercial products and 3 different lots, and they cultured them according to standard methods for each of the strains that were reported to be in the probiotic product. So by going through that, they were able to apply kind of state-of-the-art Illumina MiSeq whole genome sequencing, which then requires a lot of computational analysis to compare the data sets that they obtained and match them to reference sequencing so they could call what organisms were present in that probiotic sample. For quality control, they went to great lengths to determine what their limit of detection was, because a challenge in whole genome sequencing of complex samples, such as those from the environment or from the gut, is that it is difficult to detect a minority organism. So they determined that they could detect any microbe that was present in at least 3% of the total population. So that's their limit of detection in this assay. They also took care to determine what their rate of false positives could be, and so they set a threshold to ignore anything that was quite rare in their samples, as might occur, for example, during contamination in the laboratory laboratory steps. They also were careful to verify the results from their sequence analysis and the computational analysis using a different method, which is called a gut probe that this team has previously published through their work at the FDA. And it's essentially a microarray that has arrayed sequences from 92 different genomes and 220 different plasmids. And it uses a hybridization method so that you can take the DNA prepared from your probiotic and incubate it with this microarray, and then use detection technology to determine independently what species are present in that complex sample. So they were very careful throughout to determine the specificity of their methods and how robust, how sensitive they are as well. So what they learned is that by and large, for these 10 products, most of them did contain the species that were listed on the label, and most of the products had the microbes in the same quantity that the label indicated. However, there were a couple of exceptions. So the way they displayed their data was in Figure 2. There is a large grid that has the 10 samples arrayed in a vertical fashion, and then across the top are listed some 20 different species, I believe, that are common in these probiotic products. And then they indicate in their grid which of the products contained the species that the label indicated it did, and then independently, by using their culture-based technique as well as some PCR methods on strains isolated from pure culture, they verified using PCR the identity of those samples. And so we can see, as I mentioned, most of the products were as advertised, which is good news. They were able to detect, using this whole genome sequencing, some additional species that were present but not on the label. And most of these are harmless microbes, so there's no concern about those products being unsafe. They did find in one lot of one product, they did find a Enterococcus faecium. How do I say that?
Faecium.
Faecium. Enterococcus faecium, which you would not want to intentionally ingest. But again, it was present at very low levels and in only one of the 3 lots of this particular product. And then in another one of the products, they did detect at really low levels a Clostridium perfringens, which was at such low levels that they couldn't verify that was not a sequencing error. So they could not verify by culture that Clostridium was present. So the overall lesson here is that by using whole genome sequencing, next generation sequencing, it will be possible for companies to, once they set up this method in their lab, to in a fairly straightforward manner, verify the composition of their probiotic products. However, the authors are careful to point out that there are limits to next-gen sequencing. It doesn't tell you that the counts or the species that you detect are in fact viable in that prep. And so they currently recommend that you still also do the traditional culture method. They do say that perhaps one day we could do RNA profiling, the assumption being that if a cell is alive, it's making RNA. So if we detect RNA, you can deduce that it's alive. But currently that technology is complicated and it's expensive. So we're not yet ready to bring that to this type of quality control application.
The other issue is with these probiotic materials, they're often lyophilized to increase their shelf life. So the RNA technology would be challenging because you would have to activate the prep as you do when you ingest it in order to, if you will, wake up the bacteria that have been lyophilized in order to make them stable. Because as many of us know, that these are strict anaerobes that they are working with, or at least anaerobes, and not in the strictest sense, but they're often challenging to make shelf stable, so you don't have to have them effectively under argon gas. And how do you take a pill that's under argon gas without not violating all the other pills in the bottle? And so I think that's really the hallmark associated with this, is the authors were so careful in their sequence analysis and teaching the reader the importance of traditional culture methodology coupled with the sequencing that will enable easy lot-to-lot analysis. So sorry for interrupting, Michele.
No, we appreciate your amplifying those points. The other point they emphasize is that there is a limit of detection to next generation sequencing. So again, they won't detect anything that's, in their case, less than 3% abundance. And so it's possible that if they use only whole genome sequencing, they could miss contaminants. And some of those contaminants could be risky. So they do recommend that people still use traditional culture for that purpose. They also pointed out that whole genome sequencing, of course, is more specific, more sensitive than other tests. So the other standard method is to use PCR-based screens to identify known species. But they, in their paper, point out that there are two closely related species that the PCR test will not distinguish, but the sequencing does distinguish. And by that process, they were able to identify a couple of components of some of the products that were mislabeled as one of another really closely related species. So very easy to overlook if you're only using PCR rather than the more sensitive whole genome sequencing. But I think
that has issues as we begin to define essential substrains of microbes that are important in probiotic mixes, that the 16S technology that we traditionally use for quick and dirty PCR, if someone's there, will not be able to detect, because the 16S gene will probably be there. So you have to go and hunt for the particular remarkable gene, and whole genome sequencing is our current method for doing that. But once we begin to define the essential gene associated that gives that probiotic species its benefit to the human or host, then we'll be able to design a specific PCR primer set that will then tell us if that organism is present in the probiotic capsule at the right number. So that was another important point I think the authors taught the audience as they were reading this.
I agree. They do point out that with this technology, they could look at up to 10 products in one reaction and get the results within a week. So it is possible that the companies will move to this, and I think that's good news for the probiotic industry, that we could begin to see, a seal of approval or quality control seal on some of these products. And then just as a public service announcement, I will point out that of the 10 products, one of them performed poorly compared to the others. And so they don't name the products, but any listener who is taking probiotics could look at Figure 2 and look across the list, or actually Table 1, I believe, and Figure 2, and you'd be able to deduce if you are currently taking product H, which does not have the same. Was it product H or product G?
It was A. A has included but not found, and not on the label but found. That's all it has. It has one red and 2 yellow bars, at least in figure 2, right?
There was another product that had a contaminant and also a few of the found but not on the label. So
you can figure out which companies make these by looking at the paper?
I don't actually know how many products are out on the market, if I were taking one, I would probably look at table 2 and figure 2 and see if I could deduce whether mine checked out or not.
Yeah, you could look at what's supposed to be in it and match it up in some way, yeah.
Because table 2 does list the ingredients
Yep.
In each of the products, so I think you could deduce that. But it was impressive that by and large, the CFUs, the colony-forming units, the viable counts that the label said was in their product, by and large, those checked out, except for one of the products, product H. And again, most of the species also lined up. So I took this paper as good news.
I did.
The products out there, by and large, are what they say they are, and we can expect really higher quality control going forward.
Yep. Of course, as we have more research information on the benefits of this, right? And that'll certainly be forthcoming in the future. I think more and more people will take these, or new ones, whatever, develop. So that this will have to be done.
And I think companies will actually automate this process either by looking for specific targeted genes so that they could get away without doing the whole genome sequencing. And in fact, that was one of the limitations that the authors brought up in their discussion of the data, is that the bottleneck in this is the reduction of the whole genome sequencing data.
Yeah.
And so if you had a PCR primer set that was specific for a gene that confers the right benefit from that microbe to the host, then I think you would just need to, look for a band or look for a positive light signal. And they're probably trying to identify how to automate this for a light cycle reaction so they could see it just show up.
Yep.
But even that wouldn't turn up what are coded here as yellow, discovering additional species that were not on the label.
Yeah.
And that's the limitation of specific PCR primer sets, is you won't find things you don't want.
All right. That's very cool. I thought that would be of interest. I bet a lot of listeners take probiotics. I certainly have a few jars around my home, although I do not partake personally. And I will run them by this paper to see if I can figure out which one is which. Very interesting stuff. Thank you, Michele.
You're welcome.
We have a paper which is really, really cool, I think. And we had a listener, William, write in a letter suggesting we talk about it. And I'll read his paper later on when we're finished going through the paper. It's called Design and Synthesis of a Minimal Bacterial Genome, published in Science. And a whole lot of authors. The first author is Clyde A. Hutchison III, and the last author is J. Craig Venter, who many people will recognize. And buried there among all the authors is someone else I recognize, Hamilton O. Smith.
There are actually 2 first authors. It's hard to see the symbols, but Ray Young Chong is another first author.
That's right. Clyde and Ray Young have both contributed equally to this. This is coming from the J. Craig Venter Institute in La Jolla, as well as the National Center for Microscopy and Imaging Research at UCSD, and a company called Synthetic Genomics in La Jolla, and National Institutes of Standards and Technology in Maryland. And this has been a project going on for many, many years. And I'll start by quoting the authors who write, quote, "The genome sequence of a cell may be thought of as its operating system. It is dependent on the functions of the cell, cytoplasm, for its expression. The genome can be viewed as a piece of software." We are all just computers in the end, right?
That's right. The original biological computer, the bacterium.
And so these individuals clearly, as we'll see later, they clearly take a computational view of life. And I think we've mentioned before, the first complete cellular genomes were determined in 1985 by this group, Haemophilus influenzae, which had 1,815 genes, and Mycoplasma genitalium 525 genes. So much simpler, a reduced genome. And these mycoplasma have the smallest genomes of any autonomously replicating cells. And that's the subject of this paper. The question here is, and what has fascinating these individuals for years, what is the minimal gene set for cellular life? Or maybe you could put it, what's the smallest operating system that you could make that would have a cell life living? So initially they compared their two bacterial genomes. They came up with a core set of genes that they thought were essential, 250 or so. And then in 1999, they did transposon mutagenesis of Mycoplasma genitalium. And here you have transposable element, which hops around the genome and inserts randomly. And then you can isolate individual bacteria with insertions, and you can ask, is the gene essential or not? And they found that Mycoplasma genitalium has many non-essential genes. So in 2010, they synthesized what they call a reduced genome. They switched from genitalium to mycoses, Mycoplasma mycoses, because it grew better than the other one. What they had to do that, they developed tools to synthesize the genome. So they do that by chemical synthesis and then transplant it into another cell. They used another Mycoplasma, capriculum. And this is really cool. They call this installing the genome. It's like a plumber installing a faucet.
It's just like a CD. It's circular, just like a CD.
Installing the operating system. That's where they get it from. And this is very cool. So they synthesize, we'll talk about how they synthesize this in a minute. They can synthesize the entire genome, and then they have on it a selectable marker, and they put it into the recipient cell, and then they apply selection, and the recipient genome goes, and now you've installed a new genome. Very cool. So they were able to completely synthesize the M. mycoides genome, 1,078,809 base pairs. Basically an exact copy of the bacterial genome, except for what they call a watermark. So it's some sequence that you can use to say, yeah, this is the one that I've built, and some vectors. And they call this SYN 1.0. Version 1. This is so the computational aspects of this are so cool. SYN 1.0. So that's basically the wild type mycoplasma, which validates their whole procedure that they can put this together and put it into a cell, and then you got the cells growing. So this paper builds on that. Now they want to make a minimal genome. What's the fewest genes we can put in and get cells to grow? So they start with SYN 1.0, which is this full M. mycoides genome. And they have information from the literature, and they do a big transposon scan of the genome to identify genes that they think are non-essential and you could remove. And I'll go into this in a moment a little more. So they basically have a number of genes and say, let's get rid of these and make a synthetic genome without them. And to do this whole procedure, they divide the genome into 8 overlapping segments, which correlate with segments in the SYN 1.0 sequence. And then they can make each 1/8 segment individually. They can stick it into SYN 1.0 and see if it's functional. That way they can avoid errors, which when you make big things like this, it's huge. Over 500,000 bases, a lot of things can go wrong. So I can go through and check each of these 8 like that. They also made lots of refinements in the automated DNA synthesis. So all the DNA synthesis is automated, and it has to be error correcting because you don't want to keep introducing mistakes. And what they do is they assemble 1.4 kilobase DNA fragments from overlapping oligonucleotides, and they have ways to eliminate synthesis errors. And then they clone a bunch of these to make 7 KB cassettes. They sequence those and verify the sequence. And of course, if one isn't right, they throw it out and go back. And then they take 15 of these 7 KB cassettes to make 1/8 genome fragments. And they do this in yeast. And then they can put all 8 segments into yeast where they get assembled. And then they pull it out of yeast, amplify it, and put it into M. mycoides and do the genome installation. So this whole thing from start to finish, from the day you start making oligos to the final installation, is about 3 weeks, which is really remarkable.
Considering how much synthesis that they're doing and proofreading that's being accomplished. And they do a couple of cheats where they're having E. coli do a lot of the rolling circle replication to do some of their amplifications. But it's just a fascinating take on how to assemble a chromosome.
Right. So a little bit of information on the transposon mutagenesis is quite neat. So basically they make a big library of mycoplasma where the transposon has jumped around and you're going to get one insertion per cell, more or less. And then what you do is you take all of these insertions, you just sequence them, and you see they did 80,000 insertions. And after sequencing, they can put the genes into 3 groups. Genes which are never hit by a transposon, which means when the transposon went in, it was lethal because that bacteria didn't grow up on the plate. Then there are genes that are hit frequently, and they consider these to be non-essential. And then they have another group, what they call quasi-essential genes. When you delete them, you slow down the growth, and they had to make sure to look for slow growers to see those. So they have all this information now about all the mycoplasma genes, and then they can say, let's start taking away the ones that we think are non-essential. So they used this information. They started building these 8th genome fragments, and they put one by one into a SYN 1.0 background. And all of these worked except segment number 6, where they got very small colonies in the first 6 days. And then after that, the mycoplasma started growing quickly. And after a lot of analysis, they realized that they had mistakenly deleted a promoter in this segment, which they then fixed and got that to work.
Well, and the way they learned it is that the fast growers had sustained mutations that affected transcription termination or promoter activity, right. And they selected for natural suppressor mutations, right. Which I thought was beautiful.
So I think the anti-termination was in the previous segment, which allowed read-through right. into this one because they had deleted this promoter. Yeah, it's really cool. Mm. So they took all these 8 segments and they put them into one genome, no cells. They didn't get any cells. So back to the drawing board. And what they found out is, so there are a lot of genes where you have redundancy. You have 2 genes in the genome which have the same function. You take one away or the other away, and the bacterium will grow. But if you take both away, it doesn't grow. And they figure this must be happening on different segments. We're deleting one from one segment and one from another segment, and that's why we have this issue. So they went back and identified these genes, which, again, were functionally redundant, but were far enough away that they wouldn't have initially noticed it. And they finally got a full synthetic genome, which they call SYN 2.0, which can make a viable cell. 576 kilobase pairs, so about half the size of the original, 478 protein coding genes, 38 RNA genes, and 12 kb of vector sequence. And this mycoplasma doubles every 92 minutes versus 16 hours for the wild type mycoplasma.
Which is pretty remarkable.
It's amazing. It's really cool. But they didn't want to stop there. They wanted to keep on removing genes. So they did more transposon mutagenesis to try and get rid of more genes. And they end up with SYN 3.0, which has 438 protein coding genes, 35 RNA genes, a 531 kilobase pair genome, 531,000 bases, which they say is the smallest of any autonomously replicating cell found in nature. And the doubling time is 180 minutes, again, compared with 16 hours. And the colonies the bacteria are smaller than the full genome mycoplasma, and the colonies are polymorphic. You get a mixture of larger and smaller colonies. So an interesting thing is their discussion of what these genes are that are needed for growth. And you have to remember that they're growing these mycoplasma in rich medium in the laboratory, so they're supplying pretty much all of the small molecules that the bacteria need. So they have eliminated many genes involved in transport, catabolism, proteolysis, and a lot of metabolic processes. Those are all gone. So if you look at what's left, these 438 protein coding genes, 35 RNA genes, 41% of them are involved in expression of the genome. So genes involved in transcription, transcriptional regulation, RNA metabolism, protein synthesis, protein folding, RNA, ribosome biogenesis, ribosomal RNA modification, tRNA modification, all that stuff. 41%, a lot of the genome is dedicated to that.
Because the hardware is not enough to make a cell, right? You've got to have all that other.
That's right. 7% is involved in what they call the preservation of genome information. These are proteins involved in DNA replication and repair, DNA topology, DNA metabolism, segregation of chromosomes, and cell division. I thought that was low. I was a little surprised at that.
Yeah.
Right? I would think there would be more. But I guess you don't need a lot to do that?
I think it's because of evolution, that evolution has evolved those proteins to be so efficient. You don't need a lot of them, because if you didn't replicate your genome with high fidelity, you were depleted from the population.
But they also did not ask whether this new cell, synthetic cell, is as stable as its wild type parent. So it might be that they have deleted some of the important machinery for preserving the genome, but they just won't know that until after they culture this for a while and then go back and sequence it.
Yeah. Then 18% of the genes are involved in cell membranes, synthesis of cell membranes, lipoproteins, cofactor transport, efflux systems, protein and other transport systems. And then 17% of the genes are involved in cytosol metabolism that's going on in the cytosol. The interesting part, of course, is the 17% of the genes that have unassigned functions. These are 79 genes, and they don't really know what they're doing. So this is, if you're interested in mycoplasma, you could now start looking at these and trying to figure out what they do. Many of them are annotated, so you have some clues as to what they might do. So, for example, some of these are present in many other organisms, so they're probably important, and it would be interesting to figure out what they're doing. They make a note that many of these genes, according to the annotations, are involved in efflux systems, getting stuff out of the cell. And they say, quote, "It is somewhat disconcerting to imagine that all of these exclude or remove toxic substances." I guess it's disconcerting because they're figuring the world is so full of toxins that these bacteria have evolved to have lots of ways to get rid of them. Is that how you would read that?
I think you got to just look at their oligo machine. There's a giant waste bottle attached to the oligo synthesizing machine. And I think when you make stuff, there's always waste that has to be taken out.
Maybe.
Well, or there are other metabolic pathways that take what is the waste from one pathway and they then consume it elsewhere. So they may have inadvertently deleted some of those pathways, the recycling pathways, let's call them.
Yeah, right.
And now that cell becomes more dependent than ever on excreting substances that normally they would have recycled.
Mm-hmm. Mm-hmm.
That's kind of the optimistic interpretation.
And the natural habitat of mycoplasma is inside our cells. They're effectively obligate intracellular parasites to people. And those 79 other genes may be essential for their natural habitat, and somehow the cell has either coupled them in an operon, because they didn't necessarily comment about the fact that they have this operon structure that is carried over from how they designed their hypothetical minimal genome.
Okay. Then one experiment they did, which is pretty cool, a lot of these genes are in blocks, like for DNA repair enzymes, translation and proteins, et cetera. They're all in blocks, and they wondered, could we shuffle them around? And as you'll see, we have a letter from a listener about this. He calls this defragging the hard drive. That's pretty funny.
Or they're all frustrated flu virologists.
So they reorganized one of the modules, one of the segments, and moved some genes around and flipped them around, and really didn't make any difference. No effect on the growth of this cell, this mycoplasma. So it's kind of interesting that how they're located doesn't really matter in the genome, at least to the extent that they've looked. It could be that more extensive shuffling would make a difference, but I guess they are in blocks because that's how they were acquired originally, maybe from somewhere else, and they don't need to be in blocks. It's pretty interesting. What do you guys think about that?
You have to divorce yourself from the eukaryotic mentality that one gene, one promoter. The bacteria, because of their operon structure, they have typically one common promoter element that drives the RNA polymerase into the structural component of the operon responsible for making the messenger RNA. And then depending upon the design of the messenger RNA, they either have good or bad ribosome binding sites, and that actually controls expression. And when you make a polygenic message, depending upon the gap between genes, that RNA can fold itself into a pseudoknot that then often is either controlled by one protein involved in the system to effectively either titrate its expression or offer the cell some way of controlling the ultimate expression of a particular protein, so you always have the right concentration of protein available to it. So the polygenic nature of how microbes or how bacteria organize and control their gene expression is absent from this software metaphor where when the eukaryotic mentality is one gene gets turned on and they're not made in this operonic structure where the operon can actually be responsible for determining levels, concentrations, rate of expression, and all sorts of other very interesting behaviors in the expression of the life cycle of the microbe.
Yeah, if you have access to this paper, there's a figure 8 that shows how the gene order was reorganized. It's pretty nice because you could just look at it and say, yeah, they made some big changes and mixed up the order of these genes, and it didn't matter. I'm thinking of one similar experiment in a eukaryotic system, which was done with a virus, vesicular stomatitis virus, where there's a single initiation site for RNA synthesis at the left end of the genome. The polymerase then marches down and makes individual mRNAs. And if you change the order of the mRNAs, you mess up viral replication. And that's because there is the order of synthesis dictates how much of each mRNA is made. Mm-hmm. You have mostly the ones at the left end are the highest levels and the ones at the right end are the lowest. And if you mess them around, you mess that up. If you move them around, you mess that regulation up, and so you interfere with replication. But as you said, these have individual promoters, so it doesn't really matter that much. It's very interesting, really cool.
Another part of the paper that I really liked was they went into detail about their simple set of deletion rules that they used as they were making decisions about not what to delete, but how and where do I put the borders. Right. And I think that the 5 or 6 rules that they list here would be great for, say, a preliminary oral exam, qualifying exam for a PhD or the like.
You are an evil person.
Explain these rules. Give me examples. Why was that rule so important?
That is really good. I skipped over a lot of this. There's a lot of detail in this that we didn't go over, but that's very cool how they had to make rules and stick with them, and make sure, and if it doesn't work, you change the rules and so forth, but you have to decide, like, where do you delete? And that's what.
And what if they overlap? Exactly. It overlaps the next gene. Then what do you do?
Exactly. Yeah, it's very interesting.
And if our listeners out there are software engineers, do you guys follow similar rules in optimizing code?
Do you guys and gals?
Well, guys is gender neutral.
Apparently, it is.
My students have taught me guys is a gender neutral term.
Is that right?
Because they come in and refer to both men and women as guys.
Okay, I think I'm going to start using gals as a gender.
Gals, that is right.
We could make that argument.
Well, on TWiM, I call everyone guys, and Kathy Spindler is okay with that because she says it's gender neutral. So you could argue with her too, Michele. That's okay. So there are a lot of things you could mess with, obviously, and one thing they did was to fool around with the codon usage. So there are multiple codons for many of the amino acids, and you can change the ones that are used. And to the extent that they made some changes, they didn't have any effects, but they're talking about doing large-scale changes, in which case then you have to also change the tRNAs to match, and it'd be interesting to see if that matters. So what good is this? Well, many, many good things. You can, for example, figure out what genes are unknown but necessary and go study those. But I think what's really cool, they mentioned, you could construct a cell with any desired property as long as it works, right? So you can construct cells to make products, industrial chemicals, or useful drugs. I guess you could construct cells to degrade things if you want, and basically whatever you want. And I think this is great. I'm wondering if the people who are scared about gain of function have have read this paper and what they think about it, and whether they're going to start regulating what kind of bacteria you can make because they're scared of making something nasty. But I think the potentials are huge here.
This paper was covered in the Huffington Post, and there is already a group that is raising red flags.
Oh my gosh. I mean, this can be really, really beneficial in many, many ways, right?
But don't you think it's a little bit naive to think that you could now take this minimal cell and then, let's say, put in the genes to make immunoglobulin and assume that all the machinery that the cell will need to secrete, assemble and secrete that molecule will be in the minimal genome?
Yeah, well, that's a good point. Maybe you've taken it out in this minimal genome. You have to put it back then, right?
Right. So that's what they'll learn. What all do we need to make and export and a stable clinical product or commercial product?
I think it's not easy to do all of this. It would take a lot of time, right? And so that's why I think people who want to regulate this should just back off, because this part was the easy part, and what happens next is going to be a lot harder. But I think we will learn things as we go along.
The thing that I was most impressed with in this paper is this concept of quasi-essential.
Right.
And, that itself could also be a wonderful prelim question. What is the operational definition of a quasi-essential gene, and what attribute would you look for as you're doing a genome scan to answer a simple question of what fraction of the genome of pick your favorite organism has quasi-essential genes, and under what circumstance? Because, the genome enables the microbe to effectively experience life. When they go from a rich medium to a poor medium, and oxygen tolerance, low oxygen, high oxygen, all of these things. And, there are so many important lessons. And I think, Michele really drew our attention to it by talking about their deletion rules and helping biologists to begin to think about how we should annotate genomes and how we can begin to look at these functions, because there's a lot of information in bacterial genomes that we don't understand the context of why it's there, and more importantly, where it's located in a circular chromosome.
And also remember that this is done in the lab with rich medium. So, so although it can grow, this would never probably survive out in the wild, right? And it probably wouldn't be able to infect an animal.
And that's a counter argument to anyone concerned about biosafety. They could engineer some kind of auxotrophy so that it could not grow on its own.
That's right. It's
straightforward to do that.
Exactly. One of the things I really got to think about after I read this is, okay, in the virus world, how could we use this? Now, the viruses I've worked on most of my career have very few genes. The poliovirus encodes about 12 different proteins, and people have already tried taking each one out, and you can't get along without any of them. So it's already a very reduced organism. But we now know
evolution has already done that.
But now we've discovered viruses with huge genomes, over 1 million base pairs of DNA. And I think this, and we don't know, do we need all these in this virus? There are lots, for example, of tRNA genes and tRNA synthetase genes in those viral genomes. So this could be used to put them together and look at this. I think that would be very exciting in the virus world to do that. And I think a lot easier than this, because what you do is make the DNA and put it in a cell and see if it replicates. So I'm looking forward to that.
And this is not for the faint of heart, because, they identified a strain that they could actually swap in a genome into.
Right.
And that was a heroic effort to get that candidate strain that you could effectively replace the original or parent chromosome with the daughter chromosome that you made in a tube.
Right. I want to read this letter from William, who it's very nice. He had sent it about a week ago. Dear TWiMers.
Shall I profile the two first authors after
wonderful.
you do that, or before?
I'll read it quickly, and then you can do it afterwards. Yeah. William's right. Dear TWiMers, greetings from Berkeley, where Miracle March, water-wise, was perhaps half a miracle, but that is definitely better than none. At least we will not run out of water this summer. I found this quasi-synthetic biology result to be very interesting on several levels. First, it does not surprise me that Craig Venter was the one to do it. 30 years ago, I ran the computing group that supported the DOE Human Genome Center at LBNL when it was run by Charles Cantor. The focus was on sequencing long fragments, which was hard, but the reassembly was relatively easy. Venter went off on his own and said that shotgun, as I recall the term sequencing, was the way to go. Make lots of short fragments that can be sequenced quickly and then use statistical methods to reassemble computationally. He, of course, was right. Second, I found it fascinating that after 20 years of concerted effort, his group has built essentially from scratch the minimal life form, and in the process demonstrated that we have a very incomplete view of how life works at the most basic level. This is a quote from Atlantic article on this. However, now that they know the 30% unknowns, they can systematically tackle those genes one at a time to figure out their essential role. Even when they infer that function of some of these from looking at similar genes in other organisms where their function is known, they still end up with 17% that are essential to this minimal life form that they have no idea what they do. No idea. Surprising. Third, in a parallel experiment that didn't wind up in SYN 3.0, the scientists reorganized about an eighth of the genome to put genes with similar functions next to each other. Venter called this process defragging the genome, as one would do with a computer's hard drive. In this parallel experiment, the defragging of that section of the genome didn't seem to harm the organism. I would have thought that gene placement on the genome would be the result of some important optimization evolution, but that is apparently not the case. See the San Diego Union article. There's a nice diagram of this. Though this may be the key, quote, despite extensive reorganization, the resulting cell grew about as fast as SYN 1.0 as judged by colony size. Thus, the details of genetic organization impinge upon survival in hyper-competitive natural environments, but the finer details are apparently not critical for life. So the organization may only be non-critical in a very benign environment. Fourth, I like this, quote, the philosophy of learning basic biology by building stuff is the best bit of the SYN 3.0 story, says Drew Endy from Stanford University. Too often in biology, we end up with only data, a computer model, or a just so story. When you actually try to build something, you can't hide from your ignorance. It either works or it doesn't. Anyway, it would be interesting to hear your take on this. And he sends links to a couple of articles on this. Now, I think I might have misrepresented this defragging experiment. What he did was actually defrag the genes. The genes are all mixed up, and he put them in order of function. So the gene order of SYN 1.0, and then he went to SYN 3.0, which groups genes according to DNA repair translation and other. So he defragged it, really. I misspoke before, and that didn't affect the growth of this cell. It's really amazing. What did you learn about these individuals, Michele?
Well, they are two highly accomplished senior scientists. Ray Chung was raised in Taiwan. That's where he got his undergrad degree. And then he came to the Ohio State University for a PhD. And in 2003, joined the Venter Institute and worked there up till this past November. And then he wrote that after 20 years of basic science, he has moved to the it's ATCC. So the
American Type Culture Collection.
That's right.
Wow.
It's such a common acronym that I've forgotten what they stand for, where he is going to be working on developing products for use in synthetic biology, so a more applied projects. And he especially appreciated working on this project with Ham Smith and Clyde Hutchinson. So Clyde is the other senior scientist. He got his undergrad degree at Yale in physics, and while there, he worked with Carl Bose on bacterial spore germination. He then postdoced with Harold Morowitz before joining the faculty at UNC Chapel Hill, and there he worked on a couple mobile elements, including a phage with Marshall Edgell and line 1 elements. So these are retrotransposons, the most common in all mammals. In particular, interested in the evolution of these. He took 2 sabbaticals during his time at UNC. One was in the lab of Sanger in Cambridge, and he contributed to the first full sequencing of a DNA molecule, which was this bacteriophage X174. His second sabbatical was at Tiger Craig Venture's first company. And there he contributed to the sequencing project of the mycoplasma, the smallest genome known at the time. He also contributed with Michael Smith to developing site-directed mutagenesis in 1978 and 2005 contributed to development of global transposon mutagenesis. So he's got a really deep experience in manipulating genomes. So he is a senior scientist, senior distinguished professor at the Venture Institute in San Diego. He's also a member of the National Academy of Sciences, a fellow of the American Academy of Arts and Sciences, and also a fellow of the American Academy of Microbiology.
Yay. Wow. It must be fun to work at this institute.
Oh, gosh, yeah.
Gosh. And it's in a nice place, too, which unfortunately they don't have any water, but otherwise it's nice. It's great. Thank you.
Yeah.
Wow. This episode of TWiM is also sponsored by ASMMicrobe 2016. Registration is now open for this new meeting combining the ASM General Meeting and ICAC, and you should register before May 5th. They have early bird rates. Save some money. This brings together the General Meeting and ICAC under one roof. It's happening June 16th through 20th in Boston, Massachusetts. We'll have the best microbial scientists in the world and the full scope of microbiology. It'll also feature new venues and programs to spark discussion and interaction with your colleagues all over the campus in the dynamic exhibit and poster hall. If your work touches microbial sciences, this is the meeting you need to attend. Learn, connect, and advance at the first ASMMicrobe 2016. You can learn more and register at asm.org/ASMMicrobe, all one word. Thank ASM for their support of TWiM. I have a couple of additional emails I'd like you to hear. One is from Jenna, who writes, "Everyone at TWiM, I want to thank you again for the interest you and your colleagues have taken in our cell, host, and microbe paper. Fungal mimicry of a mammalian aminopeptidase disables innate immunity and promotes pathogenicity, which was expertly reviewed in your most recent podcast. It was wonderful to hear someone as enthused with our findings as we were." However, I just want to clarify a minor point. While it is accurate that Ilana completed most of the neutrophil work for this paper, for the past 2 years now, my own research has primarily focused on further teasing apart how blastomyces dermatitidis modulates neutrophil function by acting on other cytokines and chemokines using both neutrophil-like cell lines and peripheral primary neutrophils. Again, very much enjoyed the podcast. Appreciate the interest in our work. And Jenna is a PhD student in the Klein Lab at University of Wisconsin-Madison.
Thank you for clarifying.
Next letter from Kevin. Dear TWiM team, in episode 124, Fungal Parasites, a listener asked if quorum sensing was involved in development of antibiotic resistance. Dr. Melanie Blokesh studies how quorum signals induce competence in Vibrio cholerae. This phenomenon particularly happens in aquatic environments when V. cholerae forms biofilms on chitinous surfaces. These aquatic biofilms can incorporate multiple V. cholerae strains, and the subsequent DNA transfer between strains could promote the spread of antibiotic resistance genes. However, I was unable to identify a paper that was specifically tracking the movement of antibiotic resistance genes between species. Really enjoying the podcast. Looking forward to the next one. So we sent out a call for someone to answer that question, and Kevin does. Thank you very much. But the key here is transfer, and that is right. Being in a biofilm should facilitate transfer, and he says he doesn't know of anyone having worked on it. Our last email is from Anthony, who writes, each year we recognize World Tuberculosis Day on March 24th. This annual event commemorates the date in 1882 when Dr. Robert Koch announced his discovery of Mycobacterium tuberculosis, the bacillus that causes tuberculosis. Too many people in our country and around the world still suffer from TB. Anyone can get TB, and our current efforts to find and treat latent TB infection and TB disease are not sufficient. Misdiagnosis of TB still exists, and healthcare professionals often do not, quote, think TB, unquote. The theme of World TB Day 2016 is, quote, Unite to End TB. CDC and its domestic and international partners, including the National TB Controllers Association, Stop TB USA, and the Global Stop TB Partnership, are working together to eliminate this deadly disease, but we need your help. In some areas of Jersey City, Anthony writes, every day is Tuberculosis Day.
And to put it into perspective, a third of the planet is infected with tuberculosis.
Wow.
A third of the 7 billion people on this planet have it.
That's a lot of people.
But but many healthy people can contain it within granulomas and live a healthy life. So
that is true.
Now, I still admire a pathogen that can get into so many. Michael, do you know the virus that's in every person on Earth? Every person, every one of those 7 billion people?
It's probably one of these ones that's in urine.
It's a retrovirus. We all have retroviral DNA in our genome.
Oh, I thought you were specifically looking for a specific genus, a genus, but viruses.
I think that's so cool. Endogenous retroviruses. Now, last time in the last show, we talked about, in fact, it was Anthony who had sent us a Facebook post from Kim D'Alessandro, where she had found some interesting Petri dish art. So Anthony sends us the link to the artist's site, and her name is Ellen Thomas. And the original art in question were Petri dishes that she'd made by some kind of needlepoint, and they look like cultures. They're beautiful, absolutely beautiful.
They're fantastic.
And so Kim D'Alessandro put this on Facebook, and then we mentioned it on our podcast, and now she's come back and posted on Facebook, and she writes, "Here's how the internet works. 1, post a photo of Petri dishes by Elin Thomas. 2, photo travels around the world. 3, photo gets a mention at 44 minutes on This Week in Microbiology. 4, artist's work starts selling like hotcakes, as it should." And she puts a link to TWiM and says, listen to the whole program. You'll learn a lot. So that's cool. Kim D'Alessandro is a very famous person in her own right. So this is cool that she's publicizing cool microbiology things, including art and a podcast. So thank you, Kim, and thank you, Anthony, for bringing that up. Aren't those plates beautiful, Michele? You looked at those,
right? Oh, they
right?
are. They're just exquisite.
Beautiful. Just gorgeous.
And they look real.
Yeah, they do.
It's not, Dr. Seuss. It's
real stuff, yeah.
I don't know how someone has the eyes to be able to needlepoint at such a fine level of detail. They're absolutely beautiful.
I think youth is part of it, Michael.
Youth is part of it.
For sure. All right, that will do it for TWiM 125. You can find it at iTunes and also at microbeworld.org or microbe.tv/TWiM. And you can send us your questions and comments. We really love getting them and hearing about what you like about TWiM and what you want to know. You can send them to twim@microbe.tv. Michele Swanson is at the University of Michigan in Ann Arbor. Thank you, Michele.
Thank you both.
Michael Schmidt is at the Medical University of South Carolina, which is in Charleston. Thank you, Michael.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and in particular, Chris Condayan and Ray Ortega for all the behind-the-scenes help. Music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.